in India. And his talk is about plastic pollution and beyond. Do microbes hold the key towards a sustainable solution to this global crisis? Dr. Prasun Goswami, the floor is yours.
Okay, thank you for your nice introduction, short and crisp. So I just quickly share my presentation here. So is it visible for everyone?
Yes, it is.
Hello, everyone. Good morning over there and good evening over here. So I am Prasun Goswami. I was working at National Institute of Ocean Technology. Recently, I switched to Indian Institute of Science Education and Research as a postdoctoral researcher, which is located at Kolkata. So is it changing?
Yes, it is.
Okay. Coming to the problem of plastic pollution, if you see over here, plastic has become already the 5th most polluter in the world. And if you see the total biomass of plastic, it is already crossed the human-made mass on the earth. So in this process, entire terrestrial and marine living organisms face enormous problem due to this global crisis, that is the plastic pollution. So coming to the topic, that is what is plastic and microplastics. The plastics are what the trash is that we made in and around our environment. Of course, it is very useful material, but nowadays it causes a lot of menace out of it. And the microplastics are basically 2 types, the primary one and the secondary one. The primary microplastics is actually which is produced for industrial processes for production of like cosmetics and like face masks and even in toothpaste. Whereas the secondary microplastics generally used to get generated from the breaking down of larger plastic particles. And the crisp definition of microplastic is about any plastic particle which is below 5 millimeter in size. So why we should study about plastic pollution? So in the marine environment, zooplankton play one of the vital role. So copepod is one kind of planktonic organism. What they do is they filter out algae and other organic materials from water. And in this process, they transfer energy to the higher trophic level organisms. But if you see over here, through this process, they become susceptible to plastic ingestion. So the key question, what are the effects of plastic ingestion in this zooplankton? So my work primarily focused on understanding the ingestion part of microplastic by marine zooplankton. So if you see over here, this was published by Diksha Sharma in 2014, which showed the simulated model of global distribution of plastics and microplastic in world ocean. So if you see, the number of plastic particle increases with decreasing size, and the number of floating microplastics in world ocean is almost 6 to 7 order higher than the number of larger plastic particles. So in a nutshell, what we can say is microplastic has already created a floating continent on the world ocean. So now coming to the Indian Ocean scenario, if you see over here, in Indian Ocean, there are lots of coastal countries which produces plastics as well as microplastics. And there are a lot of riverine discharges in this region, especially here, the Ganges Brahmaputra riverine system is there. And this releases lots of plastic particles. to Indian Ocean sector, that is here in Arabian Sea and Bay of Bengal and in the Central Indian Ocean sector. But if you see over here, number of observation in the open ocean sector is very limited. However, there are already some published literature about microplastic contamination in beach or water or in biota from the coastal sector. So the highlight of this study was to point out the microplastic data on Indian Ocean is really limited. So for this, the first work what we taken was to understand the plastic pollution status in Indian Ocean. So our goal was first to map the current plastic pollution in Indian Ocean, and then to establish a baseline data for it, and then to identify the source and environmental risk for it. So, So what we did was we collected different environmental matrices like water, sediment, zooplankton, finfish, and shellfish from different locations of Indian Ocean as well as coastal areas of Indian Ocean. And we processed our samples with digestion, then density separation, and we did both microscopic and spectroscopic identification to identify microplastics in it. So the first case study, that is the plastics in food wave from the Port Blair Bay, that is in Andaman Sea, which is located in the northern Indian Ocean. We selected a coastal bay, that is Port Blair Bay, which is the adjacent to the capital city of Port Blair in Andaman Island. And we collected surface water, sediment, finfish, shellfish, zooplankton from these 6 locations in this Port Blair Bay. And what we found was high amount of microplastic bioaccumulation that was detected in zooplankton and in some commercially important fish. That is a very popular fish, carangid fish in Indian subcontinent. And we found almost up to 67 particles in this fish. So then what we got was from the regression model, organisms dimension like length and weight and feeding habit have significant influence in microplastic ingestion. So this clearly points out that microplastic ingestion changes with the organism's feeding habit as well as in different life stages. So what we observed in this work was improper waste management like fishing and shipping activities could be the possible sources of microplastic pollution in this environment. So next, we wanted to identify the hotspot of microplastic pollution in this Indian Ocean. For this, we had selected 2 locations. That is, one is in Arabian Sea, that is in Western Indian Ocean, and another is in Andaman Sea, that is in the northeastern Indian Ocean. So we collected continental shelf sediment from these 2 areas because marine sediment act as the ultimate sink of microplastics in these areas. So we analyzed microplastic in these sediments, and what we found was number of plastic particles, microplastic particles in Arabian Sea was significantly higher than the Andaman Sea, and based upon the plastic composition, that is fiber, constituted the most common shape of microplastic in both these environments. And based upon the polymer composition, acrylic, polyethylene, and nylon were the most common polymer types. And what we concluded was upwelling coupled with enhanced primary productivity may facilitate faster sinking of particles in these environments. Then we wanted to check how these plastics particles interact with plankton. For this, we had taken surface zooplankton tow from 26 locations in Arabian Sea, Central Arabian Sea, and we checked microplastic ingestion in 8 different zooplankton groups, that is copepod, ostracot, cladocerans, phasids, jellyfish, and other larvae and fish larvae. And what we found was fish larvae had the highest ingestion than other zooplankton group. So in these microscopic images, you can see this ostracods was entangled with microplastics, and this copepod was ingested microbead in it. So in surface water also, we observed a high number of microplastic pollution. So finally, what we concluded was microplastic present ubiquitously in this environment. And again, fibrous microplastics were the most abundant type, and zooplankton tend to ingest microplastic in this environment. And based upon the polymer, what we found was polyamide, polyethylene, polypropylene, PET, polystyrene, PVC. These are some of the most important polymer types. Then we wanted to identify the primary source of microplastics in this environment. So we plotted the wind current and sea surface height and salinity data. And what we found was the westerly winds used to push the microplastic from the coastal environment of the Arabian Sea to the oceanic sector. So wind is one of the major driving force over here, and the current pattern over during the sampling period revealed the occurrence of a large oceanic gyre over there, and there were some smaller eddies which formed over the southern sector, which resulted in the accumulation of plastics in the central and southern sector of Arabian Sea. Again, salinity profile and sea surface height clearly revealed intrusion of Bay of Bengal low saline water masses in the Arabian Sea, which resulted in bringing floating plastic from the coastal Bay of Bengal to the Arabian Sea. So this was clearly evidenced in our study, and based upon polymer composition, what we found was polyamide, polyethylene, and polypropylene. These are some of the common plastic types, and the polyamide major sources are like the laundry waste, which comes from the coastal areas of the Arabian Sea, and fishing gears, because these areas are very productive ecosystem, and a lot of coastal and open water fisheries takes place. And also, this is one of the busiest marine traffic area. So, the shipping gears is also another source of plastic in this environment. And based upon the polyethylene and polypropylene, what we observed was like PET bottles and other packaging materials may contribute to the plastics in these environments. So based upon this, what we understood is what is the impact of plastic in the coastal environment. So once plastic reaches from the land-based sources to the coastal environment, it starts interacting with the coastal biota as like phytoplankton, zooplankton, microbes. And also in this process, from the water environment, it used to absorb PHs and polyaromatic hydrocarbons and like other trace metals from it. And through this process, this is to accumulate and get biomagnified in the higher food chain organisms. So it is very important to study the transfer of microplastic through the marine organisms. So in the next image, what I want to show you is once plastic reaches to the coastal water, what happens is it starts interacting with marine microbes, and marine bacterial biofilm forms over there, which creates microplastics plastisphere. So as a result, this provides enormous opportunity to microbes to grow and eat, and it opens another windows of research. So for this, what we did was first we collected some plastic particles from the coastal environment of Andaman Sea, and we tried to isolate the bacterial community from it. And then from these isolates, we purify the isolates and screen for possible plastic degrading organisms. So based upon that, we selected 4 potential plastic degraders, plastic degrading microbes, which showed higher potential of up to 20% of plastic degradation. And then we incubated these organisms with culture media, and as a carbon source, we have given only LDP sheet. So we didn't add any other carbon source. And then we observed the microbial degradation process in it. So what we observed was in control, there was no growth. But whereas after 60 days, we observed attachment of microbes on it. And after 120 days, we observed lots of folds and cracks. So we wanted to check how much plastic get degraded in it. And again, through AFM studies, what we observed was increase of surface roughness, which clearly indicates that with progression in time, bacteria used to attach to plastics, and it starts breaking down the plastics in it. So this was clearly observed in our study, and based upon that, in a nutshell, what we observed was up to 47% degradation of plastics was observed under control bacteria. So this was very promising result. What we found in that, of course, we are still working on it to improve these methods to achieve further degradation process. So in a nutshell, as a take-home message, what I want to say is plastic is everywhere. So a lot of research is going on to find out plastics, but very limited research is going on so what is the impact of plastics on the marine environment? And if plastic is there, what are the solution of it? So I think it is a high time for the scientific community to start doing more and more research to find out innovative ways to develop sustainable technology and to remove plastic from the environment. So with this, I would like to thank all the organizers for providing me opportunity to present our research. And yeah, I think I will end over here. Thank you. And you can also always read our papers for further information. Thank you very much.
Yeah, thanks a lot for this great talk, Prasun. Very interesting, very exciting. Do you have any questions from the audience? You can either unmute yourself and ask directly, or you can write it in the chat, as you prefer. Yes, there's one question. Please go ahead.
Yeah, thank you to the presenter. Your emphasis, you put too much emphasis on wind as a source for the pollution in the aquatic environment, specifically the Indian Ocean. Can you then you talk about fishing gears? I wanted you to talk on some land-based activities affecting or some land-based activities, maybe runoff could carry plastic or could carry microplastic to the Indian Ocean, or there's no runoff activities in our area, or is there any all of land-based activities that can move microplastic to the aquatic environment?
Prasun, did you hear the question? I don't see him online anymore.
No, he's fallen out of the list.
Oh.
We might have to save questions for Prasun until he
Yes.
reconnects.
Exactly. I'm sorry for that. So maybe you can re-ask your question in the end, please. I'm sorry for that. So in that case,
I was saying in his presentation, he talks about wind as a source for moving microplastic to the Indian Ocean. He also talks about fishing gears. So is there any other maybe idea can he elaborate on concerning maybe runoff or some land-based activities that cause microplastic to move to the aquatic environment? Is there any idea he can share with us on that? Any land-based activities that can move microplastic to the aquatic environment?
yeah, I'm sorry because Prasun, the presenter is not here. So I wanted to ask you to ask your question in the final discussion after the second talk. Because
hopefully he will come back.
Exactly. Hopefully he will come back. I'm sorry for this. So in that case, we move on to the second speaker, which is Ms. Diksha Sharma from the CSIR-National Institute of Oceanography, Biological Oceanography Division in India. And her talk title is Impacts of Zinc and Copper Enrichment on the Ocean Acidification Scenario on a Phytoplankton Community from a Tropical Upwelling System, the Arabian Sea. The floor is yours.
Okay, thank you. Thank you so much for the introduction, Pascal. Can I share my screen now?
Yes, please.
Yeah. Yeah. Is it visible?
Yes, it is.
Okay. I've started the slideshow. Okay.
Yes, works perfectly.
So shall I start?
Please go ahead.
Yeah. So hello, everybody. And it was a very good presentation, which Dr. Prasun has presented. So I'll try my level best to maintain the standard over here. So, okay, so this is Diksha Sharma, and I have submitted my thesis in December 2021, and I'm expecting my final defense to be soon, maybe in another week or so. So this is part of my PhD work, and I have done my PhD under Dr. Hemanti Biswas from National Institute of Oceanography, India. And I'm going to present on this topic, which Pascal has already introduced. So coming to the introduction part, in general, phytoplankton are the single cell microorganisms which present as suspended or floating within the sunlit area of aquatic ecosystem. So they constitute the base of marine food chain and buffer our climate by fixing approximately 30 to 50 billion metric ton of carbon annually. So in the ocean, these oxygenic phytoplankton comprises approximately 1 to 2% of the global plant biomass, but fixes 30 to 50 billion metric ton of carbon, which comprises approximately 40% of the global primary production. So in the ocean, as we know that oceans are the major sink of carbon dioxide and responsible for the extraction of 1/3 of the carbon dioxide from the atmosphere. So this net transfer of carbon dioxide from the atmosphere to the ocean and then to the sediment where it is getting sequestered for centuries is carried out by biological carbon pump, as you can see on the screen. So this biological carbon pump transfers approximately 11 to 15 gigaton of carbon annually, and without it, the atmospheric carbon dioxide concentration would be 400 ppm higher than the present day scenario. So this BCP, biological carbon pump, comprises of phytoplankton, the primary producers, zooplankton, and the fishes. So these BCP, the oxygenic photosynthesis by phytoplankton lowers the partial pressure of carbon dioxide in the upper ocean, and which enhances further the carbon dioxide dissolution from the atmosphere. And this leads to the formation of particulate organic carbon in the epipelagic region. And the bacteria and zooplankton further work on this particulate organic carbon and converted into fecal pellets and aggregate formation, and transfer it to the mesopelagic and then to the bathypelagic by sinking. And that's how the sequestration of carbon is happening. So as these carbon dioxide and phytoplankton, they are so interlinked. So it's very important to understand how carbon dioxide is impacting the ocean. So as it's a very general thing that atmospheric carbon dioxide is increasing at an alarm rate, and it's already increased above 400 ppm. So how it is impacting the ocean, as the increase in the atmospheric carbon dioxide is impacting the surface ocean carbonate system. So what is the mechanism behind it? When after the dissolution of carbon dioxide in the ocean, it forms carbonic acid, which further dissociate into bicarbonate and carbonate and hydrogen ion. So as the dissolved inorganic carbon present in the ocean comprises of dissolved CO2, which is less than 1%, carbonate, which is less than 10%, and bicarbonate approximately 90%. So when there is an increase in the dissolution of atmospheric carbon dioxide in the open surface ocean, the equilibrium of this carbonate system is shifting towards the dissolved carbon dioxide and bicarbonate, and the carbonate and the pH is decreasing with the increase in the dissolution of carbon dioxide, as you can see from this pH. And it has been predicted that it will lower the pH from 8 to 7.8 with the 50% reduction in the carbonate concentration in the by the end of this century. So that's this lowering of pH because of the increase in the dissolution of carbon dioxide is called ocean acidification. And why this study is very important in this particular area, because this area is considered as a natural laboratory for ocean acidification study. The Arabian is a perennial source of carbon dioxide because it is the most extremely productive oceanic region, which has many other interesting features like open ocean upwelling, coastal upwelling, convecting mixing, and one of the intense oxygen minimum zone. So there are two monsoons which we experience over years. So the strong low-level jet, fin letter jet, passes across the Arabian Sea parallel to the Oman coast. So the north side of the fin letter jet experiences open ocean upwelling because of the positive wind stress curve, and the south side experiences downwelling. So, and this wind reverses during the winter monsoon. So as the wind in the southwest monsoon was towards the westerly direction, in the winter monsoon, it's from the northeast wind. This old and dry wind comes from the Indian subcontinent, and which drives the convective mixing. So because of these processes, the Arabian Sea experiences remain productive most of the time of the year. And as Arabian Sea is facing a combination of multiple stressors like ocean acidification, higher SST, trace metal pollution, and atmospheric aerosol influx. So the combined impact of ocean acidification along with these variables bear considerable capacity to impact the ecosystem functioning, and rather than ocean acidification alone. And it is quite plausible that the marine phytoplankton inhabiting this particular area will also show a highly diverse and complex responses to these combinations. So let's focus on the trace metal first. So as from the schematic diagram, you can see that as the trace metal, as the name suggests, is required in a very trace amount for the physiological activity and the microbial and the growth of the phytoplankton. There are trace metals like copper, iron, cobalt, manganese, which are very important for the physiology of phytoplankton. Suppose let's take iron. These trace metals serves as a cofactor in several enzymes as well as biomolecules like proteins. So suppose if we are taking iron performs several functions in phytoplankton, like it is required for the chlorophyll synthesis, it is required for the uptake and assimilation of nitrogen, which is the second abundant micronutrient required for the microbial growth. And it is also required for the electron transportation during the process of photosynthesis and respiration. Similar, zinc is also present as a cofactor in carbonic anhydrase, which plays a crucial role in the carbon concentrating mechanism. And as well as it also participates participates in the hydrolysis of organic phosphate ester to orthophosphate with the help of alkaline phosphatase. So these trace metals plays a very significant role in the microbial growth. But as the name suggests, it required in a trace amount. But when they are in the elevated concentration, they are proved to be deleterious for the growth of phytoplankton. So why? Because the toxicity of trace metal depends on the binding of these trace metal to the coordination sites of the coordination sites of the enzyme present in the phytoplankton. So these coordination sites are non-specific. So there is so it's become very it is impossible to understand. It is impossible to understand which metal is getting attached to the coordination site because it's a non-specific coordination site. So generally these coordination sites get attached to the essential metal only, but there are certain times when the non-essential metal get displaced. These essential metal get displaced with the non-essential metal, and it inhibits the particular metabolic activity because it doesn't have that chemical characteristic, and that leads to the toxicity of these trace metal for that particular process. So as these trace metals are present in different oxidative states, so seawater pH plays a very crucial role in their solubility and bioavailability in the aquatic ecosystem. So as these this carbonate and hydroxide anions form a very strong complexes with trace metals, divalent and trivalent, and these anions are going to get reduced due to the ocean acidification in the future. So it will lead to the high fraction of free metal ion in the aquatic ecosystem. As you can see from this graph by Milero et al., the copper free ion concentration is going to increase in the future because there is a competition between the organic ligand and hydrogen ion and metal ion to bind to the organic ligand. So as the hydrogen ion concentration is going to increase, it will bind to the organic ligand, and which will increase the fraction of free metal ion. So as this is very interesting bubble chart. As you can see over here, why I want to work on the combination of multiple stressor, because this is a meta-analysis carried by Gunderson et al. In 2016, where they have showed that how many studies are carried out on the combination of stressors and their impact on the phytoplankton. So the most common was the salinity and the temperature, and then followed by temperature and acidification. But interestingly, as you can see, there is no single study on the combination of acidification and trace metal and their impact on the phytoplankton physiology and community structure. Recently, there are 2 or 3 studies, but from Arabian Sea, this will be the first study to assess the impact, combined impact of these 2 stressors on the phytoplankton physiology and how the community is getting restructured. So, which brings to the work which I have done in my PhD. These are the location where I have done, I've carried out 8 experiments, but today we are going to focus only on these 2 experiments, which I carried out in the southwestern coast of India during the upwelling, during the southwestern monsoon, which experiences upwelling. So this is the experimental design. We have conducted 2 consecutive experiments, one with the copper and carbon dioxide, and the other is the zinc and carbon dioxide. And over here, it is important to note that we didn't do this experiment on the natural phytoplankton community. We have initiated the artificial bloom and modified the natural phytoplankton community to avoid the impact of grazer, so that we can get the exact idea how these stressors will affect the physiology and the community structure of the phytoplankton of this community. And this is the experimental design. I will not get into it right now. If you have any questions about it, we can have a discussion after this presentation. This is the initial parameters for both the experiment. The ambient was around 400 microatmosphere in both the experiment, and high CO2 was around 1000 microatmosphere. And the initial concentration of copper and zinc was, copper was approximately 11 nanomole, and zinc was 4 nanomole. This is the light intensity manipulation, which we have done while setting up the experiment. So coming to the results part. So first, we're going to focus on the carbon dioxide and zinc enrichment on phytoplankton assemblage. So as you can see, the first graph is showing the cell density, and it's clearly visible that under high CO2, there was a significant increase. This red arrow are showing the statistical significant changes, differences among the among the treatment. So this red, under high CO2, there was a significant increase in the cell density. And let's focus on the carbon dioxide right now, carbon dioxide, effect of carbon dioxide. So as this bubble chart shows the community structure of this particular experiment, as you can see, there was a significant increase in the concentration, abundance of ketoceros, which is a centric diatom. So there are certain studies like Atal et al. 2008, where they have also noticed that under the high CO2 concentration, the small pinnate species is getting replaced with the bigger centric species. As you can see from here, just a second. As you can see over here, the Nischia is getting replaced with the ketoceros. So why is it happening? Because this centric diatom must have got some energetic benefiting and outcompeted the small pinnate species. And the other thing is, like, the impact of increasing CO2 on a phytoplankton depends on their DIC acquisition. As we all know that the main carboxylating enzyme that is Rubisco has a very low vicinity towards the carbon dioxide as compared to oxygen. So the half saturation constant of carbon dioxide in Rubisco is around 40 micromole per kg, and the dissolved carbon dioxide in the world ocean is around 10 to 12 micromole per kg, which limits the optimum photosynthesis. So and there is another thing, that the concentration gradient of the CO2 across the cell wall is high, so which also leads to the loss of cellular carbon dioxide via diffusion. So to attain maximum photosynthesis, they have developed carbon concentrating mechanism where they are increasing the carbon dioxide concentration in the vicinity of the Rubisco by taking the CO2 by taking dissolved inorganic carbon in the form of bicarbonate at the cost of energy. So when there is ample amount of carbon dioxide provided under the high CO2 environment, they down-regulate the CCM and reallocate that energy for another cellular functions. So this could be the reason why these bigger centric diatom outcompete the small diatom and flourished in this experiment. Coming to the zinc addition, the very interesting thing which I want to show it over here, in the cell density, there was a significant drop in the cell density when we have added zinc under ocean acidification scenario. But as the biomass indicator did not show any significant change, but per cell chlorophyll and per cell POC under high CO2 plus zinc showed a significant enhancement. So as I mentioned earlier in the previous slides, the binding of the metal ion depends on the concentration of hydrogen ion present in the system. So what is happening, because of the increase in the hydrogen ion concentration, there is an increase in the zinc ion concentration, which is ultimately inhibiting the calcium uptake in the calcium ATPase activity, which is required for the cell division process. So what is happening over here, there is an uncoupling between the cell division and the photosynthetic rate, which leads to the accumulation of photosynthate in the cells and enlargement of the cells as they are not able to divide. So this could be the plausible reason why the per cell chlorophyll and per cell POC is high and cell number is less under high CO2 plus zinc concentration. And there are studies which have showed the similar trend, like Stauber and Florence, where they have added 1 nanomole of zinc in the culture of Nischia clostridium, and the cell division became completely half, but there was no effect on the photosynthesis. Even when they have increased the zinc concentration up to 7 nanomole, the similar trend they have noticed, no impact on the photosynthesis, but the cell division drastically reduced. So this could be the reason for this kind of trend which we have noticed in this study. Coming to the second experiment, carbon dioxide and copper enrichment. Unlike zinc and CO2, we have not seen any significant change in the photosynthetic performance and the cell growth rate of this experiment, as there is no statistical difference within the treatment. But albeit of the fact that it did not show any significant change in the cell growth and photosynthetic performance, community composition did change and showed a change when we have added copper in it. So as you can see, there are like, these are the species which dominated in this experiment, and Leptocylindronicia showed a significant decrease when we have added copper in both the CO2 level. But Pseudonicia interestingly showed enhancement in all the copper level in both ambient as well as high CO2, which means that they must be getting benefit when they are in the copper enriched environment. So there are studies like Lelong in 2012, where they have showed there are certain species of Pseudonicia which are known to be a toxin producing species because they produce domoic acid, and this domoic acid is a copper chelator, which means that the more the concentration of copper, there will be a more prevalence of this particular species. And there are other studies like Sun et al. 2011, where they have noticed under high CO2 concentration and copper concentration, there is a flourishing of domoic acid producing strain of Pseudonicia. So these observations clearly conclude this thing, that under copper pollution in the in copper polluted area under ocean acidification and nutrient limited condition, these toxigenic species gonna prevail, which can have a severe impact on the trophic levels, different trophic level, as well as on the community structure, as well as on the carbon sequestration. One more thing I would like to mention over here, this work has been carried out in the southwest coast of India during the upwelling time, and there was a study by Rationale et al. 2009, where they have found double the concentration of copper in the same location where we have performed the experiment. Like we have noticed 11 nanomole of initial concentration of copper, and they have approximately observed around 20 nanomole of copper. So this could be the plausible reason why these phytoplankton species must be acclimatized to that particular high concentration of copper, and as it was in the upwelled area, they must be acclimatized to the high concentration of CO2 also, because a significant amount of nutrient and CO2 comes to the surface water during the, maybe because of that reason, we have not seen any significant change in the biomass as well as in the growth rate. So coming to the conclusion part, as we have not seen any inhibitory as well as stimulatory impact of carbon dioxide in the phytoplankton community in both the experiment, because of the upwelling, as the phytoplankton inhibiting this area is experience upwelling. The centric diatom may gain benefit from the increasing CO2. Zinc did not show any significant change in the ambient, but found to be extremely deleterious for cell division under ocean acidification scenario. Zinc was more deleterious as compared to copper. Some phytoplankton like toxin producing pinnate diatom like Pseudonicia may proliferate under copper enrichment and ocean acidification scenario, and future increase in copper pollution along with ocean acidification may facilitate the harmful algal bloom, which may adversely impact the ecosystem, and such community may have a considerable impact on the trophic transfer, carbon export plus carbon cycling in this region. So these are the papers which we have published from this work, and two are under revision, and this is the published paper. And thank you so much.
Thanks a lot. This was a great talk. Are there any questions in the audience? Now we have kind of a common, let's say, discussion. So questions for both speakers are very much welcome. There's one question in the chat. I think it's for you, Diksha. How do you count the cell?
Yeah, we have used, as the experimental community was, we have initiated the artificial bloom. So it was dominated by diatoms. So we collected the samples from the experiment, and then we have used the microscopic analysis to count the cells.
Thank you. And there, happy to see you back, Prasun. There were two questions actually for you.
Okay.
And there was one which was written. One sec. It was from Nazir Ahmed. How do you identify microplastics using epifluorescence microscopy?
Yeah. That's a great question, actually. Some of the microplastic they used to emit fluorescence. So once we excite under the under the fluorescence, it used to emit its natural fluorescence. Certain fluorescent stains, which I didn't use in this study, but there are fluorescent stains like even DAPI and Nile red, with these people used to identify microplastics.
Thank you for this answer. There was a question of Anvesha. Do you want to ask yourself or should I read? Okay, it's in the chat. What kind of plastic show fluorescence, the colored ones?
Yeah, it's again, Indonesia, it's a great question. There are plastics like polypropylene and again acrylic that is mostly used in fibers. They use under the UV light or even under the fluorescence. Yeah.
Thank you for that. There was one question which was asked in person. I'm sorry, I will probably pronounce it wrong, but it was from Kanzualism. Maybe you can ask it again, or you want to write it in the chat. It was for Prasun.
Yeah, I just want to know, I'm from the Institute for Environmental Protection. We noticed that so many land-based activities causing pollution in the aquatic environment, specifically plastic. And most of the industrial commercial activities, the plastic might either travel through runoff water or through different means to enter the aquatic environment. And in your deliberation, you talked about weighing and fishing gears. So I was thinking, per your experience, can you share with us further any other means through land-based activities that microplastic can enter the aquatic environment?
Yeah, I didn't follow in the last. Can you please repeat your last part?
Yazi, can you elaborate further? Can you talk more on some of the land-based human activities that causing microplastic?
Yeah. One is primary one, and primary one, it comes from like interchange. These primary microplastics are produced and used like wave, wind, tide, everything. Like it starts breaking down. And also there are biological processes like microbial degradation. So what happens? This larger plastic tends to break down into the smaller plastics in a decreasing number. So it generates microplastics. So once plastics reaches on the surface water, from the land-based like landfill or from any seawater treatment plant, a sewage treatment plant, it tend to float and with the water flow and reaches to the coastal areas or even in the oceanic environment boundary. So once to the long distance and tides everything is the ultimate source of plastics, but it is hypothetically proven that there sources. One is land-based, and another is like a fishing industry and shipping, the like municipal wastewater, even everything, solid waste, everything.
So part of that answer, I think, at least on my end, was very choppy. So I'm not sure if we got the whole story, but there might have been some more connection issue.
Okay.
Yeah.
Okay.
Are there any other questions for either speaker? If somebody is still thinking, maybe in the meantime, I can call for speakers for the next sessions. We are always looking for people who would like to share their research in this seminar series. I will put the link for the application in the chat. And the second thing I would like to stress is please share Amplifying Voices in your network. I think the more people are joining this, the better, because this is the intention of the series, to share research among the whole community. So please spread the word. Are there any other questions? If not, I'm sure if you think about anything later, I'm sure you can contact the speakers via Twitter or via email. And yes, I will put this in the chat, but I would like to thank both speakers for their talks and for the interesting discussions. And I'm looking forward to see